In anatomy and neuroscience, a dorsal root ganglion or spinal ganglion is a noodle on a dorsal root of the spine that contains cell bodies of nerve cells called neurons that carry signals from sensory organs towards the appropriate integration center. Nerves that carry signals towards the central nervous system, brain, or spinal cord are known as afferent nerves. Unique unipolar structure. The axons of dorsal root ganglion neurons are known as afferents. In the peripheral nervous system, afferents refer to the axons and relay sensory information into the central nervous system. The brain and the spinal cord. These neurons are pseudo-unipolar type, meaning that they have an axon with two branches that act as a single axon, often referred to be as a distal process and the proximal process. Note, the neuron can consist of three parts. 1. Dendrite, that receives information and relays it to the soma or cell body. 2. Soma, the cell body of the neuron. 3. Axon, which relays information from the soma. If a neuron that dendrites receives information from another neuron's axons at the synapse, and the axon sends information to the next neuron's dendrites, even though the dendrite may be covered with myelin. Unlike the majority of neurons found in the central nervous system, an action potentially in dorsal root ganglion neuron may initiate the distal process in the periphery, bypass the cell body, and continue to propagate along the proximal process until reaching the synaptic terminal in the dorsal horn of the spinal cord. Distal section. The distal section of the axon may be either a bare nerve ending or encapsulated by a structure that helps relay specific information to the nerve. For example, a Meissner corpuscle or Pacinian corpuscle may encapsulate the nerve ending, rendering the distal process sensitive of the mechanical stimulation, such as stroking or vibration, respectively. Location. The dorsal root ganglia lie along the vertebral column by the spine. Embryology. The dorsal root ganglia develops in the embryo-form neural crest cells, not neural tube. Hence, the spinal ganglia can be regarded as gray matter of the spinal cord that became translocated to the periphery. Mechanosensitive channels. The nerve ending of dorsal root ganglion neurons have a variety of sensor receptors that are activated by mechanical, thermal, chemical, and noxious stimuli. In these sensory neurons, a group of ion channels thought to be responsible of somatosensory transduction have been identified. Compressing of the dorsal root ganglion by a mechanical stimulus lowers the voltage threshold needed to evoke a response and cause actions potentially to be fired. This firing may event persist after the removal of the stimulus. Two distinct types of mechanosensitive ion channels have been found in the dorsal root ganglion neurons. The two channels are broadly classified as either high threshold HT or low threshold. As their name suggests, they have different thresholds as well as different sensitive to pressure. There is a cationic channels whose activates appear to be regulated by the proper functioning of the cytoskeleton and cytoskeleton associated protein. The presence of these channels in the dorsal root ganglion give reasons to believe that the other sensory neurons may contain them as well. High threshold mechanosensitive channels. HT channels have a possible role in nociception. These channels are found predominantly in smaller sensory neurons in the dorsal root ganglion cells and are activated by higher pressures. Two attributes that are characteristic of nociceptors. Also, threshold of HT channels was lower in the presence of PGE2, a compound that sensitizes neurons to mechanical stimuli and mechanical hyperglycia, which further supports our role for HT channels in the transduction of mechanical stimuli into nociceptive neuron signals.